Hey guys, welcome to Anatomy and Physiology of the Cells. Now, first comment, a lot of this is a rehash of material from Biology 1. Alright, it just is, but I feel like it is pertinent because later on when we get to things like how nervous tissue really functions, how you are capable of transmitting a nervous signal from one spot in the body to the other, you need to have a really good grasp of types of ion channels and intercellular connectivity and all that fun stuff. So I go through all this stuff as a primer so that we can better understand real cellular function as time progresses. So with that in mind, let's roll through it. Okay, so cells and cell theory. It wasn't always apparent that we were made of cells. Actually, it wasn't until a few hundred years ago that we really came up with the concept. And this wasn't finalized until, Schleiden and Schwann came up with their unified cell theory, okay? And I find this fascinating. I'm going to run through it with you just because it's interesting. All organisms being composed of cells and cellular products. Again, in the distant past, this was almost heretical. We didn't have this full grasp. You can see your arm and think, wow, that's not cells, that's an arm. But people didn't have a real understanding of what microscopic things were. So this was really cutting-edge science for the time. Cells being the basic structural and functional units of life, so all living things are composed of cells and cell products. This basically is why we say that viruses are non-living, because viruses are acellular. Activities of cells manifest themselves as organismal activity. That is to say that tissues, being groups of specific types of cells that do a specific job, that sort of leans into the concept of a tissue. Cell activities reflect their structural components, same basic concept, and then of course life being propagated through cellular activity or reproduction. Again, you have to think about this as flying in the face of the thoughts of modern time, or of the time, I should say. This sort of takes all the magic out of how life is made. So cell theory had its ups and downs for a little while. Alright, good enough. So here's our good old-fashioned animal cell. Now I'm going to run through the parts and pieces with you real quick, and then we're going to go into grandiose amounts of detail. What we have here is a nucleus with its nucleolus. Nucleolus is where ribosomes tend to be made. The nucleus houses the DNA. Here is rough endoplasmic reticulum. We say it's rough because it has ribosomes attached to it. This is smooth endoplasmic reticulum. Here's a Golgi body, sort of adjacent to the rough ER. So you kind of go nucleus to rough ER at the Golgi. Here are mitochondria, which provide the energy for the cell in the form of ATP. We've got lysosomes that house some digestive enzymes. And then, of course, we have this cellular membrane. And the membrane is not a solid structure per se. It is a fluid mosaic. Things are constantly moving around in and around it. And this cell membrane has openings through which things can pass. And we'll talk about how those openings work in a little while. Alright, here we go. The plasma membrane, the outer surface of the cell, will be our first conversation. Alright, so the cell membrane is mostly lipids, about 98%, the vast majority being phospholipids, these unsaturated phospholipids. So this is your phospholipid bilayer. You can see the phosphate head and the lipid tails. These are those fatty acid tails. And what will happen is the phosphate heads kind of stick together because they are all polar. And then the tails kind of stick together because they are all non-polar. So the heads are polar, they are hydrophilic, they're water-loving. Heads, water-loving. Tails, hydrophobic, they are water-fearing, they are non-polar. So what you end up with is this double layer. Your cell membrane is known as a phospholipid bilayer, and it looks something like this. But what I want you to notice immediately is that there's way more going on here than just the phospholipid bilayer. There's a lot of other things here. So we've got like cholesterol all throughout this. Cholesterol is going to help keep the membrane fluid when the cell gets cool, or it'll help to solidify the membrane when the cell gets too hot, basically keeping the cell from losing its function. There are, of course, lots of glycolipids and general sugars found on the outside of this cell membrane. These sugars are very important because they function as molecular markers. I probably have glycocalyx. Yeah, I've got the glycocalyx on here. These sugars on the outer surface of the membrane form a glycocalyx. This is a sugar coating of the cell. In essence, what this does is it forms up a method of what we call self, non-self recognition. Okay, self non-self-recognition. This basically allows your white blood cells to detect your cells versus like a bacterial cell or something. So a bacterial cell will have different external cellular markers than yours, so your white blood cells can kind of feel the outer surface of a cell and identify if that's you or if that's something else. Okay, so these sugars on the outside assist with this. And also there are occasionally some what we call saturated phospholipids that are referred to as lipid rafts. Saturated phospholipids stick together far more strongly. They form almost a solid platform, and we think that these may play a role in a process we call endocytosis, which we'll be describing in just a second. Integral proteins and peripheral proteins. So the membrane is mostly lipids, but most of the mass of the membrane actually comes from these big honking proteins that are found associated with the membrane. And these proteins come in two flavors. There are integral proteins. Integral proteins span the entire length of the membrane. And then there are peripheral proteins. Peripheral proteins will be found on the border of the membrane only. So peripheral protein, integral protein. Yeah. Peripheral proteins tend to be associated with cytoskeleton, that's a fact. Tend to help with cell division. Yeah, that's all true. Alright, far more important from my perspective are the integral proteins. Okay, this slide contains information that I am going to be just harping on pretty much for the whole rest of the time we're in this class. Okay, this is incredibly, incredibly important, so please pay attention. Some integral proteins function as receptors. These will bind to, for instance, like a hormone on the outer surface and then cause an internal change to the cell, cause something to happen. Maybe this is insulin and this is a liver cell. When that insulin hormone molecule binds to the receptor on the liver, it triggers an internal change which causes the liver to store sugars. You know, these receptors are incredibly important. Some of these can be enzymes that can break things down or build things up. Ion channels and gated ion channels are just super important. Okay, super duper important. Ion channels allow a free flow of materials through the cell. These have to be incredibly small things like single ions, like a sodium ion or a potassium ion or a chloride ion. And individual water molecules tend to be able to pass through this as well. These ion channels form up in red blood cells to make what we call aquaporins. And these are little holes through which water can flow. All right, red blood cells are very osmotically active, and they use these aquaporins to keep balanced with the plasma around them. There are also gated ion channels, and gated ion channels just, whew, they are important, man. They really, really, really matter. Let me explain how. So, if you are hearing the words I'm saying right now, the reason you are hearing these words is because I'm vibrating the air, which is being picked up by my camera, that is being incorporated into a video, which eventually makes it to your computer and comes out your speakers as sound. That sound coming out of your speakers vibrates the air, which is then picked up by your ear. That sound vibration comes and hits your tympanic membrane, shakes that tympanic membrane, vibrates your three ear bones called the malleus, cecus, and stapes, which in turn vibrates the oval window of your cochlea. Yes, this is all true. And as it vibrates the oval window of your cochlea, just like that, a piston-like motion, what will happen is it sends vibrations through the cochlea that bend these special cells in your ear called your hair cells, and when those hair cells bend, it cracks open gated ion channels that stimulate the neurons in that structure. When I touch this table, I'm opening gated ion channels in neurons on my fingertips so I can feel my table. When I see that screen, the rods and cones, my visual receptors in my eyes, are cracking open gated ion channels, which in turn stimulate nerves, which crack open their gated ion channels. Gated ion channels are the deciders. Okay? If they open, message is gone. If they close, message is stopped. I can attach an electrode to a muscle in your body and send a shock through it. And what the zap does, what that shock does, is it forces your cells to open their gated ion channels and to become stimulated. I can make your muscles twitch by sending a shock through them. And the reason for that is because the shock will crack open their gated ion channels and stimulate for flexion. These are so important. So important. All of our physical activities, it's all based off of gated ion channels and their operation. So these are just so important. Their capacity to open and close, man, like the reason your heart's beating right now is because gated ion channels are opening and closing, allowing that heart to beat. It's so important. All right, we've already kind of talked about cellular identity markers. These bear sugars, which allow us for communication between cells, or allow your white blood cells to do what we call non-self recognition, being able to detect your cells versus foreign cells. And then, of course, there are cellular adhesion molecules, or what we call CAMs. Cellular adhesion molecules basically allow a cell to attach to an area and hang to it. They're like little cell hands. Your white blood cells are very good at this. They do a process called diapetesis, where your white blood cells will get into your tissues and kind of crawl around looking for problems. And the way that they can attach and crawl around is by using SAMs. Apparently we're talking about surface-to-air missiles. By using CAMs, these cellular adhesion molecules. Okay, perfect. Now, in the cell, we have a cytoskeleton. Just like your body has a skeleton, your cells have a skeleton. And in this, the cytoskeleton, we have three parts that we're going to discuss. The microfilaments, the intermediate filaments, and microtubules. Microfilaments are made out of little actin molecules, these actin protein subunits in essence. And what these will do is they will allow for cellular motility. Cells can move a little bit. Some cells can move a lot, like your muscle cells can really move. But other cells in your body can move a little bit as well. They can kind of shrink up a little or kind of expand, and they do this through the use of microfilaments. And then there are intermediate filaments. All right, intermediate filaments are associated with desmosomes. All right, desmosomes, and what they do is these are kind of like guy wires on a power pole, or a better way to put it is they're like wires that hold something together. Okay. They're like rebar and concrete in essence. So what these will do is they will attach two cells together so that if you pull against those two cells, the desmosomes make those two cells function as one unit. So intermediate filaments kind of link all the cells in a tissue together in some cases so that that tissue is very strong as a unit. Okay, strong as a unit. They're strong if you pull against them, but they have no compressive strength. They're like ropes. They're like ropes. That'll do. Microtubules, on the other hand, are completely different. Microtubules are like tent poles that hold up a tent. Okay? They are a rigid structure. Microtubules are a rigid structure. They have compressive strength. They are like big old power poles that hold the cell open when it's being compressed. All right, big, strong structures. And not only that, but microtubules also associated with motor proteins to carry and move things around within the cell. You can think of microtubules and microtubule tracts as the subway within your cells. When your cells need to move something from one spot to another, they rely upon microtubule tracts for that movement. Okay. For instance, a person with Parkinson's disease, one of the problems with Parkinson's disease that leads to nerves dying is their microtubule tracts break down and they can't transport materials around the cell anymore, so the cells die. Okay, microtubules are very important. Alright, so we're changing gear here a little bit. What we're going to talk about now are modifications to the cell surface which are protein associated. And not, for that matter. Alright, let's just go with it as we see it. Hang on. Weird. Let's just do microvilli and then we'll go from there. Alright, these are microvilli. I kind of put this slide in out of place, but that's okay. We'll just go with it. There are microvilli. Microvilli are little surface modulations on a cell membrane, as you can see here. Little finger-like projections. These are non-motile. These are not cilia. These are microscopic compared to cilia. Cilia are not as small as this. These are tiny, tiny, tiny little structures. When you look at them under the highest magnification we would have in a lab, it just makes the surface of the cell look kind of hazy. Okay, you can't really see them. But what these do is they vastly increase the surface area of the cell. And the more surface area there is at the top here, the better these cells will be at absorbing nutrients. Okay, so a cell with microvilli has lots of surface area, and because it has lots of surface area, it assists in absorption. So you find these in, for instance, like in the small intestine. Okay, the small intestine is excellent for absorptive capacities, and the reason the small intestine is really good at absorption is because the cells there have microvilli. Okay, microvilli, very good for absorption. All right, next we're going to talk about tight junctions, desmosomes, and gap junctions. These are all modifications on a theme of proteins that are cell membrane associated. All right. Do we have this lined up correctly, I hope? So this should be tight junctions, this should be desmosomes, and this should be gap junction. Good. Okay, so tight junctions first. The idea behind a tight junction is that these are areas where the cell membrane has two peripheral proteins that kind of link the cell together, or actually integral proteins that link cells together. So they kind of clamp these cells together. So here's one cell membrane, here's a different cell's membrane. The tight junctions form a junction which is tight. Okay, the essence of it is the materials can't meander between cells because the junction's so darn tight. Then there are desmosomes. All right, desmosomes are associated with intermediate filaments, as we described previously, and these intermediate filaments attach to desmosomes. The essence is that the desmosomes will kind of clamp the cell membrane together, and then you have these radiating intermediate filaments that link to other desmosomes. If I go back to here, you can see this. Desmosome, intermediate filaments, desmosome, intermediate filaments. They link those cells together structurally so that all the cells are linked and they're very strong if you pull against them. Okay? And then there are, of course, gap junctions. Gap junctions are junctions with a gap in them, as the name implies. What we have here are two integral proteins with an opening between the two that's called a connection, amazingly. What this basically does is it allows the cytoplasm of this cell to flow through into the cytoplasm of this cell. The cytoplasm of this cell to flow through into the cytoplasm of this cell. And the idea is that if I stimulate this cell, that stimulus flows through and into the adjacent cell, stimulating that cell, which flows through and stimulates the adjacent cell. When cells are connected via gap junctions, they function chemically as one individual cell. Your heart is a really good example of this. All of your heart muscle, let me try again. All of your heart muscle is interlinked with gap junctions, so that if one area of the heart muscle begins to contract, the whole heart contracts. Okay? Which is very important for what's referred to as its pacemaking capacity. And you'll do that when you get to anatomy too. That'll work. Then there are cilia. Cilia are structural modifications, as you can see here. You can see them with a microscope. They are hair-like, they stick up, and these cilia can be non-modal, or alternatively they can be modal. So the best example I can give you of this is in your respiratory passages. Basically, when you breathe in, you get a lot of debris from the air around you, and your tracheal passages have respiratory cilia that are mobile. And what they do is they constantly are beating and pushing the debris you breathe in up out of your respiratory passages. And you know this, because every now and again you're going to be sitting there and you're going to clear your throat. You're going to go, and the reason you clear your throat is because that debris collects at your epiglottis. It's the structure right at the top of your throat, and gets dumped over into your esophagus. So when you cough, what you're doing is you're coughing that junk up out of your trachea into your esophagus so you can swallow it down. It gets a hydrochloric acid bath and it can't hurt you. So you can breathe in all kinds of terrible pathogens, but they are not able to hurt you because they're caught in your respiratory mucus, and your modal respiratory cilia beat them out of your system. And then, of course, there are non-modal cilia. Non-modal cilia function as sensory receptors. When I was talking about how you hear things earlier, and I said you have hair cells that are bent, yeah, we're talking about non-modal cilia. You have non-modal cilia in your ears that detect motion. That's probably the best way I can describe it to you. And you'll be seeing that when we do special senses at the end of the semester. It's pretty cool, actually. And then, of course, there are flagella. The only real flagella in the mammalian system is a flagellum that's on a sperm, and these are way bigger than a cilia. And whereas a cilia just does this power stroke, recovery stroke, power stroke, recovery stroke. Flagella, man, it beats like a snake's tail. It allows for a swimming-like motion, which is how we propel sperm to egg. All right, so they are quite different in reality. And I'll say this too, when we're kind of on the subject. Mammalian lineage flagella are really different from flagella you see in the microbial world. Like a bacterium, for example. A lot of bacteria have flagella, and they're a whole different animal. Completely different. I would love to have this conversation with you, but, it's not as important. All right, membrane transport. So we need to get into passive mechanisms and active mechanisms of transit. The idea is this. Passive transport mechanisms like diffusion and osmosis, facilitated diffusion, these are non-energy requiring passive mechanisms. Really small things can move from an area of high concentration to an area of low concentration, from high concentration to low concentration, via passive mechanisms. They just kind of flow from high concentration to low concentration. Real small stuff can do that with great ease. And then active transport. This is how we move either very large things or how we move things from low concentration into concentration. Like if your cell needs to pump a specific ion into a cell and concentrate it in incredibly high amounts in that cell, it's going to use active transit. That's referred to as being either primary active transport or vesicular transport. Again, the variation here being that these consume ATP. Simple diffusion. So, simple diffusion, quite frankly, is the movement of something from a high concentration to a low concentration, just flowing down that concentration gradient, no big deal. There's a lot of things that affect this. Temperature, so as temperatures go up, the amount of movement goes up. The molecular weight, so larger things tend to move much slower, small things tend to move much faster. This is life. Steepness of the concentration gradient, the more concentrated something is, the faster it diffuses away. Membrane surface area, so if you want to have materials flow into a cell more quickly, you have to have more surface area on that cell. That's that principle that we talked about with microvilli, with the intestinal tract. And then, of course, membrane permeability. So a cell that has a membrane which is more permeable will have diffusion happen through it much faster. This is actually what happens with insulin and cellular glucose. So when your blood glucose levels are too high and you release the hormone insulin, what that insulin actually does is it causes your cell membranes to become more permeable to glucose. And then there's osmosis. Osmosis is simply the movement of water from high concentration to low concentration. The idea is that water chases solutes. This is the principle behind how your kidneys function. Having fluid moving through your very salty kidneys, you end up pulling fluid out. Anyway, it's way too complicated for us to get into here, but osmosis is quite frankly the movement of water from high concentration to low concentration. And then, of course, we must talk about general tonicity. So we had spoke previously about how your red blood cells are very active osmotically because they contain protein units called aquaporins. Do I even have aquaporin on here anywhere? Just trust me when I say your red blood cells have aquaporins. And the idea is this is all stuff that you should have been doing in Biology 1, but if you place a cell, let's do isotonic first. You place a cell into an isotonic solution, osmotically balanced is what that means. Isotonic means osmotically balanced. So the amount of water in versus the amount of water out is equal, so that cell will maintain its perfect little biconcave pink donut shape, a good old-fashioned red blood cell. Alternatively, if you place a cell into a hypotonic solution, when I think about hypotonic, think about enlarging. Basically, the cell is placed into a solution where there's more water outside the cell than there is on the inside. There's a really high solute concentration inside the cell, so water just rushes in and it fills it like a balloon, and it can actually lead to what we call lysis taking place. It can pop it like a balloon with hydrostatic pressure. And then alternatively there is hypertonic, a hypertonic solution. Place a red blood cell into a oh boy, this is hitting me hard. If you place a red blood cell into a hypertonic solution, what that means is that there's more water inside the cell than there is on the outside. Imagine this cell being placed into hardcore salt water. Well, what's going to happen? The water in the cell is going to rush out to try to equalize with the salt. This is osmosis. And the cell is going to crenate. It's going to shrivel up. And if you look at these little spikes sticking off the surface, those spikes are microtubules. Remember we said they're like tent poles trying to hold the cell open? Well, there they are, and you can see them. Okay, microtubules. Okay, and then of course there is active transport. And the first part we're going to get into is primary active transport. What you got to think about here is this: we're using ATP to move things either into concentration or to move very large substances or substances that otherwise struggle to get across the cell membrane. Okay? Quite simply, straightforward, first part of this, sodium-potassium pumps. What a sodium-potassium pump does is it pumps sodium and potassium, one to one side of the cell membrane and the other to the opposing side of the cell membrane. They're going to pump sodium, let's see, I believe sodium gets pumped out and potassium gets pumped in. So they stay at really high concentrations, all the sodium on the outside and all the potassium on the inside, don't quote me on that. And then, given certain circumstances, those are allowed to very quickly trade places to some degree via gated ion channels. So what we'll do here with these sodium-potassium pumps is you'll use ATP as a power source to grab sodium and pump that sodium in one direction. And then you'll grab potassium and pump that potassium in the opposing direction. A pumping action, pumping things into concentration, that is primary active transport. And then there's vesicular transport. All right, vesicular transport is broken down into what we call endocytosis or exocytosis. Endocytosis is bringing things into the cell. Exocytosis is letting things out of the cell. And we can further break that down just a little bit. Endocytosis can be broken into phagocytosis, pinocytosis, or receptor-mediated endocytosis. Phagocytosis is to engulf large particles. Pinocytosis is to bring in fluid. Cell drinking is what we call this. And then there's receptor-mediated endocytosis. And here is the bit that I want you to be familiar with. Of the three of these, this is what you really need to know. Receptor-mediated endocytosis is kind of famous for being part of the placenta. So during gestation of a fetus, the placenta has to pick up very specific ions or very specific nutritive molecules or what have you. And to do this, it has receptors on the placenta, and when those molecules are present, it forms a vesicle and pulls them in to deliver that to the growing fetus. Okay, it does this in a very special way by using what is called a clathrin-coated vesicle or a clathrin pit. Okay, clathrin is a protein that sits typically, as we were saying previously, on a lipid raft, and it'll just form a coating on the surface of the cell. So when the receptors pick up that material is there that it needs to take up, the clathrin changes shape and it forms a vesicle, and it snaps off, so you get this clathrin-coated vesicle. And then what happens next is very fascinating. Once the vesicle is made, the clathrin unfolds, goes back to the cell surface, reattaches, and new receptors are set up, and it does the whole process again. So this clathrin is very important for receptor-mediated endocytosis, which is very important for how the placenta works. And I think that's cool. Okay, I think that's fascinating. So I think that's something you should be aware of. And also something you should be aware of, another neat protein interaction here, is with exocytosis. Okay, in exocytosis, you don't just send a vesicle to some random place on a cell for that cell to then release that vesicle. That is not the way this works. The vesicle has to move up via a microtubule tract. It's kind of like a factory, okay? A factory has a loading dock, an area where things can be loaded onto trucks and carried away. And in the same way, your cells have very specific areas for exocytosis. And those areas are known to us because they have snare proteins, okay? Snare proteins. What these proteins do is they will interact with the vesicle that's going to be released from the cell, and they bind together, as you can see here, and basically unzip the cell membrane to let whatever product it is out of the cell which is there to be released. Alright, so you can't have receptor-mediated endocytosis without good clathrin pits, and you can't have exocytosis without good snare proteins. Okay, the snare proteins and clathrin, they're very important. One for endocytosis, one for exocytosis. Man, Marie Curie. Boy, we should talk about this. So I've been skipping around the people, but boy, Madame Curie is important. So Marie Curie is probably top 5 smartest humans of all time. Alright, and I don't say that lightly. She's probably 1 or 2. Very super intelligent. She has 2 Nobel Prizes, and they're in different fields of science. Like a normal super genius human will work their entire life, and before they die, they get a Nobel Prize for their work on one subject. Marie Curie got 2 freaking Nobel Prizes in different fields when she was relatively young. She died young because she was running a lot of radiological experiments without radiation shielding. So the assumption is she died from cancer brought on by the radiation. And not only that, but she was a real hard ass, for lack of better terminology. It's noted that Madame Curie would like run X-rays for soldiers on battlefields. Like, it's complete madness. I mean, she was really amazing. So an absolute genius with no fear. There's a famous quote actually. Nothing is to be feared. It's only to be understood, or something like that. You should go do some reading on Madame Curie, man. She was hardcore. All right. Let's see. Into the cytoplasm. So what I want you to think about here is that the cytoplasm is the soup. Okay? Everything the cell has to do, it's got to have the building blocks to do those jobs, and the building blocks do any of the jobs the cell has to do. Well, that comes from the cytoplasm, folks. The cytoplasm is the fluid in which all things pass within the cell. The cytoplasm is the fluid in which all of the building blocks, the monomeric subunits of proteins, for example, are found. Like, it's all in there. Okay, it's all in there. Stuff floats around, and it doesn't float around randomly. It's held in place, but the cytoplasm is the transit unit. Okay, stuff flows through this. Within the cytoplasm, you find mitochondria. Mitochondria are very important because they produce the ATP via cellular respiration that our cells have to have for all of their basic processes. Mitochondria, by making this ATP, are very important. They're derivatives of a special type of bacterium, actually. And if you look inside the mitochondria, they actually have their own unique bacterial DNA sequence. It doesn't match human DNA. It's a pretty neat thing, man. Mitochondria, the powerhouse of the cell. They make ATP. The nucleus, man, it's such a neat structural set. So the nucleus is not just a nucleus. The nucleus has a lot of parts and pieces. And some cells don't just have one nucleus, some cells have multiple, and some cells don't have any. So let's talk about it. A good example of a cell that doesn't have a nucleus is your red blood cells. That's why they have a relatively short shelf life compared to others. In other words, they don't live that long because they don't have a nucleus. In other words, if the nucleus is the area where you store all the genetics, so and when I say genetics, I mean all the code for making proteins. A cell which has no nucleus can't make protein, and when it becomes damaged, it's destroyed. That's red blood cells. Red blood cells lack a nucleus in their mature stage. And then you have cells like your skeletal muscle cells that have lots of nuclei. See, skeletal muscle cells can be huge, man. They're really long, and because they're so long, it's not really effective to just have a single nucleus. So they have multiple nuclei all throughout the cell so that they can make protein wherever is necessary. Your muscle cells are very active, so they require this. What's next? 3 regions. Alright, the 3 regions of the nucleus are the nuclear envelope, the nucleoli, and chromatin. Okay, and this is very simple. Is this the next slide? Yeah, okay. Which is on the next slides. The nuclear envelope is important because it has very small, what are called nuclear pores. Alright, you can see the pore here. And what's important about the nuclear pores is their size. You see, they're too small for DNA to get out of. DNA molecules are quite large. DNA molecules cannot escape the nucleus. They are housed in the nucleus exclusively. So what you have to do is you have to make an intermediate of a DNA molecule, a copy of a gene to make protein outside of the nucleus, which is where we make protein. We make proteins out in the cytoplasm. So what do? You make a copy of a gene from your DNA, and we call that an RNA molecule, RNA, that can then leave via the nuclear pores because the RNA molecules are small enough to fit through and go out into like the rough endoplasmic reticulum, for example, for protein synthesis. So the nuclear pores are important because they can allow things in and out, but they keep the DNA where it's supposed to be. Yes. Perfect. Nucleoli, as seen here, these are little dots inside the nucleus. You can see these under light microscope analyzing. They are very clearly visible. And nucleoli are areas where we're making ribosomes. So the ribosomes are actually made in the nucleus, and then they fit into the rough ER, typically, where we make proteins. Ribosomes not being protein themselves. Ribosomes are nucleic acid. And then, of course, there is chromatin. Now, chromatin is a buzz term for DNA. It's a little complicated. If I asked you what DNA is, you'd probably say a double helix. And you're right, DNA is in the form of a double helix, but there's a lot more going on here than you probably realize, and I'm going to run through that with you. A lot of time when you think about DNA, you think about these X-shaped chromosomes that we see just prior to cellular division. Checking my time. All right, just prior to cellular division, and this is not the way that DNA normally is within your cells. Normally, DNA in your cells is in this, or in your nuclei, I should say, is in this really diffuse form where it's loosely held and it's just forming up into a big ball of multiple chromosomes that we call the chromatin of the nucleus. All right, so when you see a nucleus and you see the nucleoli around in there, there's just DNA everywhere. It's uncoiled. It's still a double helix, but it's uncoiled, and you cannot see individual chromosomes. Now, that chromatin contains, about 30% DNA. Most of it, actually, is in the form of what are called histone proteins. Okay, histone proteins are important because they assist the DNA in maintaining a regular structure. They kind of keep things marginally organized, do the histones, which is important. Yeah. Chromosomes being a very tightly coiled form of chromatin. So chromosomes will form up in their tightly coiled formats like this just prior to cell division. Mitosis. The endoplasmic reticulum. So the ER comes in two flavors. There's rough ER and there's smooth ER. Rough ER is very straightforward. This is going to be where proteins get made. The rough ER is rough because it's studded with ribosomes. It's got ribosomes studded to it. So you can see the ribosomes here. This is rough endoplasmic reticulum. This is the area of protein synthesis. Smooth ER is very different. Okay, smooth ER is highly variable. Like in the liver, it's going to be used for detoxification of certain chemicals. In your muscles, the smooth ER is specialized to store calcium. It just depends. Like, it's going to be used for manufacturing steroid hormones, making cholesterol if your cells need cholesterol. I mean, you name it, right? The smooth ER can do a lot of different jobs, whereas rough ER is quite focused. Okay, ribosomes, which we've already talked about, are important for the manufacture of proteins. And then, of course, there's the Golgi body, or the Golgi apparatus. Now, the Golgi body, quite simplistically, is a packaging center. Proteins that are made, or kind of proteins that are made in the rough endoplasmic reticulum are all floated into the Golgi, where they get finalized, and then they're all floated out of the cell, or they're released from the Golgi into the cell to do some sort of job. Imagine it like this. The rough ER is where you make your primary strand of a protein. That's all floated via a vesicle into the Golgi, and then the Golgi does the final work to it. It's an assembly line, folks. The way I want you to think about this is it is an assembly line. The nucleus contains the DNA, which is the code for making a protein. That gets offloaded into the rough endoplasmic reticulum where the primary strand of that protein is made. That gets offloaded via a vesicle into the Golgi body where everything is put in its final usable form, and then that is either released from the cell or released into the cell to do some sort of job. It is a factory. Exocytosis, okay? It's a factory for making proteins. That's what your cells do. That's what your cells are. Okay, don't overthink it. Cells are not that fancy. It's a factory for making proteins. Perfect. Cholesterol and various other functions, synthesizing lipids, smooth ER. Yeah. Alright, so let's change gears here a little bit, and we will talk about protein synthesis. Man, here's Watson and Crick, Maurice Wilkins and Rosalind Franklin. These four folks are very important for the discovery of the nature of DNA. Watson and Crick get most of the love because Watson and Crick sounds nice. But these two folks were also very, very important for discovering the nature of DNA. And so let's talk about DNA and its generalized structure and function. What we have here with DNA is our general nucleotide structure. This is going to be a sugar phosphate with an attached nitrogenous base unit, being a purine or a pyrimidine, which we talked about previously here being adenine. So sugar phosphate, sugar phosphate, sugar phosphate, and a nitrogenous base. So there are 5 different nitrogenous bases that you are familiar with at this stage. The purines being adenine and guanine, double rings. Purines, adenine and guanine, and the pyrimidines being cytosine, thymine, and uracil. Now, very important nuance, in DNA, thymine is found. In RNA, uracil replaces thymine. You don't find thymine in RNA. RNA will contain uracil in the place of thymine. That's the only real variation that's worthy of mention at this stage. What I want you to notice here, this is our good old-fashioned double helix of DNA. And if you unwind that DNA double helix, you get something like this. Very, very important for you to know is that the sides of this sugar, phosphate, sugar, phosphate, sugar, phosphate backbone, if you will, for your double helix is covalently bonded and incredibly strong. Whereas the central portion with these nitrogenous bases, adenine to thymine, cytosine to guanine, these are going to be hydrogen bound together. And hydrogen bonds are weak attractions due to positivity and negativity between structures within these molecules. Basically, these are just weak attractions. So if you want to open up a DNA molecule to replicate it, what you're going to do is you're going to pop open these hydrogen bonds. All right? So the hydrogen bonds are a little weaker. The covalent bonds are very, very strong. Now, further, you need to know who binds to who. And what I mean by that is you need to understand that guanine binds to cytosine and cytosine binds to guanine, and alternatively that adenine binds to thymine and thymine binds to adenine. It's very easy to play this game. If I were to put on your test a sequence of nucleotides and I said, what would the corresponding DNA sequence look like? It would be that adenine binds to thymine, DNA. Guanine to cytosine, adenine to thymine, cytosine to guanine, thymine to adenine, adenine to thymine, guanine to cytosine. It's easy. Alternatively, if I said, what would the corresponding RNA molecule look like? Okay, if we're making a strand of RNA, like a messenger RNA strand, it would be adenine, not to thymine, but adenine to uracil. Because in RNA, uracil replaces thymine. So adenine to uracil, guanine to cytosine, adenine to uracil, what have you. I might also put on your exam, I might say something like, which of these two is RNA? And you would know that this is RNA because it contains uracil. So these are things you need to understand about the structure of DNA. Covalent bonds, hydrogen bonds, who binds to who, sugars and phosphates and nitrogenous bases, purines and pyrimidines. Uh-oh. Alright, we've already played this game. We know where we're at. We know what histones are. We know how the molecule coils. You have seen this X-shaped chromosome before. You know a little bit about it at this stage, I hope. These are sister chromatids. These would be identical. Before mitosis occurs, we double our DNA complement to get the cell ready to split. And when this happens, you get these X-shaped sister chromatids. I'm going over this quickly because prerequisite to this class is Biology 1. You're supposed to understand general mitotic concepts at this point. So I'm just kind of going over the top. Right? Going over the top. So we have these sister chromatids, and they are identical to one another. And that takes us to the cellular life cycle. You know that mitosis is the splitting of a cell to make two brand new cells. Okay? I am not going to go over mitosis in any kind of real detail here, because you're supposed to have done it in Biology 1. You should be familiar with it. But what you probably didn't get as much of in Biology 1 is what's happening during interphase. My opinion is that interphase does not get enough love when you are doing your basic sciences, and there's so much important stuff happening here that's important as we progress through anatomy and physiology that I feel like talking a little bit more about interphase is worthy of our consideration. So let's talk about interphase. Interphase is broken down into 3 steps, and you need to be familiar with these 3 steps. They are the G1, S, and G2 phases. The G1 phase is where cells do their job. A liver cell that's being a liver cell, it's going to hang out in the G1 phase. A heart cell that's being a heart cell, it's hanging out in G1. Nerve cells that are doing nerve cell things, they're in the G1 phase of interphase. If that cell ever tips into the S phase, it's going mitotic. This is the avalanche coming down the mountain. If it ever goes into the S phase, it's coming down into mitosis. That cell's going to divide. You have a lot of cells in your body, specifically like your nerve cells, which are non-mitotic. They get into the G1 phase, and we oftentimes will refer to it as a G0 phase, but regardless, they get into this G1 phase, and they just sit there for the entire duration of your life cycle. So you got nerve cells in your brain right now that have been there since you were in the uterus, and they're going to be there when you die, hopefully. All right, so they sit there forever. They're very long-lived. But other cells, like your skin cells, for example, they are quite mitotic. And what will happen is they'll move from G1 to the S phase. S phase means synthesis. What are we synthesizing? We're synthesizing new DNA. Very importantly, pay attention, we're synthesizing new histones. New histones in the S phase. Synthesizing means to make new DNA, new histones, to get that cell ready to divide. We're doubling our DNA complement here, getting the cell prepared. And then the G2 phase is basically just finalizing all the arguments to get the cell completely ready to go through mitosis. And that's when you hit that mitotic phase and crank through and make 2 new cells. Interphase is where the cell spends the majority of its time. Mitosis, by comparison, is just a very quick process. We need to talk about some enzymes. And you know these are enzymes because look at the way they're named. DNA helicase. Anything that ends in ASE, folks, if it ends in ASE, it's an enzyme. Okay, so we got DNA helicase, DNA polymerase, and I guess that's all we're going to talk about for this particular set. So let's do that now. DNA helicase is very special because it unwinds the DNA double helix. It splits it in twain and allows for DNA polymerase to come in and connect, one on each end of this leg, so 1 leg, 2 legs, DNA polymerase, 2 separate ones, and it's going to start laying down new nucleotide base pairs. So if I want to split a DNA molecule and make 2 brand new ones from the ashes, what I'm going to do is I'm going to take DNA helicase, crank that sucker open into 2 parts, and then DNA polymerase is going to come in and lay down the base pairs. So helicase does the unwinding, polymerase lays down new polymeric subunits to make 2 brand new DNA strands. Yeah, perfect. And that takes us here. This is incredibly important for us. Your DNA contains many chromosomes. Your DNA contains many chromosomes. Chromosomes contain many genes. Genes determine protein. Okay, a gene is basically an area of a DNA molecule that codes for a single protein. So your DNA has a lot of chromosomes. Each of those chromosomes contains a lot of genes. Those genes, each individually code for a single protein, and that's where we're headed here, folks. We're going to talk about protein synthesis in all of its nuance and glory. The idea is that proteins are incredibly diverse, and they just do so much for us. It is so important. For instance, like keratin is a very supportive protein structure for your skin, keeps your skin strong. Enzymes like amylase help us break down starch. All right, this is super important for our digestive process. Hemoglobin, which carries oxygen within our blood, allows for us to transport oxygen around through our tissues. Hemoglobin is a protein. Antibodies, insulin, which is a hormone, modal proteins like actin and myosin. Man, let me tell you, proteins are just incredibly important for us. So let's go through, talk about some proteins and protein synthesis, and get this finished up. On to the concept at hand. So cells do indeed have a molecular chain of command. That is a fact, and it goes as follows. None of this should be a surprise to you at this stage. DNA is located in the nucleus of your cells. Why can't it leave? Because the nuclear pores are too small for the DNA to escape. The DNA is always in the nucleus of a eukaryotic cell. RNA polymerase, ASE, what kind of molecule is that? That is an enzyme. RNA polymerase transcribes DNA genes into an RNA molecule. Okay? What RNA polymerase does is it takes a gene and it makes an RNA molecule using it as a template. The DNA is unharmed in this. RNA molecules are made by RNA polymerase, leaving the DNA unharmed. RNA then moves out into the cytoplasm where proteins are made. Specifically, the RNA molecule moves out of the nucleus into the rough endoplasmic reticulum because that's where the ribosomes are, and ribosomes are the site of protein synthesis. So, 2 key terms you need to know. Those are transcription and translation. And you need to understand that DNA is the code for how to make proteins, but the intermediate molecule in this is RNA. What happens is this. Take home message. DNA is transcribed into RNA. RNA is translated into a protein. Okay? DNA transcription occurs in the nucleus. The RNA molecule then leaves to be translated out in the cytoplasm, specifically in the rough endoplasmic reticulum, into a functioning protein. Now, it's not all that simple. There's actually 3 different types of RNA, and they are as follows. There is messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA, transfer RNA, and ribosomal RNA. What do? Messenger RNA is the product of RNA polymerase. Messenger RNA is a copy of a gene. Messenger RNA is what's transcribed. Messenger RNA is transcribed using RNA polymerase in the nucleus to form a messenger RNA molecule that can then leave the nucleus and go out into the rough endoplasmic reticulum where translation occurs. This brings up our next victim, transfer RNA. Okay, transfer RNA is basically just a molecule which carries amino acids. This will have 2 very important locations that you need to be aware of. These are an amino acid binding site, so this is going to hold amino acids, and what's called an anticodon site. Now, I'm not going to deep dive into how codons and anticodons work, but the gist of it is that you need to understand, in the English language, we've got a word here, and each letter means a little something sound-wise in a word. You could go and look at hieroglyphics, and it's like ideas, words are ideas. It's kind of, there are Chinese characters that get these really ornate characters and have lots of different meanings in a single character. DNA is a codon system. It's in groups of 3. Adenine, adenine, guanine means something. Guanine, cytosine, thymine means something. They're in triplet codes. Bam, bam, bam. 3, 3, 3. And the idea is that this anticodon site fits into one of these triplets. Okay? So this is like adenine, adenine, guanine. That's going to fit into, on an RNA molecule, uracil, uracil, cytosine. Okay? This anticodon, the anticodon site basically can anchor to a messenger RNA molecule and impart amino acids to a growing polypeptide strand, as you're going to see in mere moments. Take-home messages: there's an anticodon site, there's an amino acid binding site and an anticodon site on a messenger RNA molecule. Transfer RNA molecule. The messenger RNA is a copy of a gene. The transfer RNA has a codon site or an anticodon site and an amino acid binding site. And then last but not least, we have a ribosomal RNA molecule. Now, ribosomal RNA simply is a ribosome. Okay, this is just a ribosome. Think of ribosomal RNA as just the 2 parts. Ribosomes are a top half and a bottom half, and they kind of clamp together on the messenger RNA strand. Like this. Bottom half, top half. That's the ribosome. See there in yellow and green? That's the ribosome. This strand here, that, folks, that is messenger RNA. These little blue things coming in here, connecting to the messenger RNA strand, connecting. These are transfer RNAs, and they're bringing in individual amino acids that's leading to the growth of a polypeptide. This is a polypeptide. That is a bunch of amino acids binding to one another covalently, but because it's protein, we call it a peptide bond. We're making a single primary strand of a polypeptide. This is not finalized. This is not a functioning protein. It's a polypeptide. That can then be offloaded to the Golgi body, wrapped up in a special way using hydrogen bonds, and that will be a functioning protein, like insulin. Okay? So that's how this is done. What we're going to have is our messenger RNA molecule. This is translation occurring. Our messenger RNA molecule here. We have our ribosome. We have our transfer RNAs, and we're making a polypeptide. Now, quick comment. A messenger RNA strand isn't just going to have a single ribosome on it, and it's not going to be just used once. What we have in reality is what's called a polyribosome. We'll have a messenger RNA strand, and every square inch of that thing, every usable amount of area, is going to be lined up, running down the length of this messenger RNA strand, making protein, or making polypeptides, as it were. All right? It's called a polyribosome. The messenger RNA strand is not destroyed. It's used over and over and over and over and over and over and over again. This, folks, this is how we make protein. Transcription into a messenger RNA strand, translation into a primary strand of a polypeptide that can then be turned into, via hydrogen bonds, secondary structures, tertiary structures, quaternary structures, alpha helices, beta pleated sheets, that kind of thing, turned into a functioning protein. So this process is very fast and very accurate. We can measure this in bacteria, for example. A polypeptide strand can grow at a rate of about 20 amino acids per second, and very seldom are there mistakes. Basically, the polymerase molecules can kind of spell check. Okay, they can check their work and make sure there weren't any screw-ups. So as they are working, they're spell checking and making sure there's no problems. So the chance of making malformed proteins from a good RNA strand, very rare. Okay, very, very rare indeed. And this whole process we've been describing is incredibly energetic. It takes lots of energy to do all of this crazy process. It takes lots and lots of energy. Let's see. Take-home messages. Information in a gene is ultimately used for making proteins. The DNA of each gene contains information to make a single polypeptide. That's a fact. So one gene codes for a single protein. Each chromosome has many genes, about 25,000 total in you. Yeah, man. Transcription, translation, RNAs and intermediate, peptide bonds between amino acids. Man, let me tell you, I think that's got it.